Hello, folks. So, just to kick off this session, the first question I have is, do you know what a Tullgren funnel is? Tullgren.
No. Sometimes called the proper name is the Tullgren Baermann. Anybody know what? Any takers? Not a brace. Yeah, comes under ecology. So it does. Does that help? No. No, that doesn't help if I say ecology. Anyway, it comes under ecology. So, any takers? Anybody know what it's for? Still people coming. Resounding silence means that it's a new thing for you. Okay. We'll get going with it anyway. Comes as a kit. You don't have to have it as a kit. It can come. This is it. So you got a funnel. You got a rod kind of piece there, and you got a light. Does that make any? Does that help you? Or is that still weird? Okay, so we got a light piece. And we get, hey, there we go. So you've got an actual funnel. You've got a rim here, and the rim has a gauze in it. That's not a particularly fine one. If you buy the set, you don't have to. You can have different size gauzes. Right, just go for the medium. Okay, so obviously there's if you've got different size gauzes, you're trying to screen something. But what are we screening? So retort stand. I got a boss connected there. So normally you might need this rod here, but you can put it straight onto the retort stand. But the ones here, I think, are pre-metric. So we'll do that one. Now, what I'm looking for, I was just checking to see, was there another one of these retort rods, they're called. Okay, so obviously you plug it in to get a light, but what is it that we're shining the light on? Okay, so we got that eliminated. No, still nobody. Might as well bring it down. It's about there. What are we trying to do here? Okay. Luke, what do you reckon? Something to do with ecology. And just turn off your.
Sorry, what was that? My Wi-Fi is just a bit slow.
Why, you're looking it up under Google?
Oh no, the connection here is
I know what you're messing with your head. Anybody? Rob? Any thoughts?
Didn't hear the question, sorry.
Any thoughts on this? What is it? I know it's a Tullgren funnel, but what's it for? And believe it or not, that's a junior cert question. But it's Leaving Cert Biology as well. And it comes under ecology. And you're probably thinking, and I know ecology is not your favorite thing. Because at the first day when I asked you about things that you like or don't like, some people did put ecology. And I amazed with that, because that's when you get kids outside to collect stuff. So we put some of the stuff we collect and we put it in this, we shine a light on it, and something comes out, but it's quite specific.
Soil.
Yeah, it's soil. It's soil in the gauze, but what comes out? And this is the specific thing. So it's for checking to see what's in soil. All right. So Tullgren funnel is for separating arthropods out of soil. So it is. There's one little bit that's missing down the bottom, preservative, so that when the little guys drop out of the soil and they fall down, they drop into the preservative. And there's a reason for that. It's because when you set this up, you mightn't come back to it for a day or two. It's sitting at the lab and you're too busy doing other teachery things. Okay. And then what happens is, of course, they just shrivel up and turn to dust. You come back and you look, oh, just soil dust. And you think there's nothing there, but yes, because you missed them. Okay. So, and also if, there's other things, that go and eat them in the meantime and so on. So anyway, so you need a little beaker of preservative. What preservative do we often use? What do I use, for example, when I have the jars of animals and I'm preserving in there? In the old days, it was formaldehyde. And formaldehyde is a carcinogen. So we try to not use that nowadays. So we use a thing called vinosetol. Okay, so vinosetol. Okay, and you buy that, you just mix it with water. And what happens is you can use that as a preservative. Okay, now it's a preservative. There are some things that you still need formaldehyde for because it's a fixative. But generally speaking, with arthropods from soil, you can extract out the arthropods and put them in the phenoxyetol, and it'll do. So you're not mucking around with the flipping formaldehyde. Okay, you can avoid that. Okay. So, so we're trying to check what organisms are in soil, because should there be organisms in soil is the question. Should there be, should you be finding stuff in soil? And what's that an indication of? So what you should be doing is you should be seeing lots of living things, because soil is a living matrix. Okay, it's something that contributes to all the crops and things that grow and so on. Okay, and the soil, it's also a distinctive ecosystem. So you could do your whole ecology section just on soil alone. All right, because it's an ecosystem in itself. You could keep earthworms. You could get students to be looking at the earthworms in a wormery. Okay, we have something like that here, but I'll set that up next time and you can see how to do that. But so we got this. So we're going to set up this. We're going to take some soil from the base of a pot plant, because it's fresh and it's damp, and we're going to see if there's any things living in it. Okay, and it's from topsoil. Yeah. Okay. Use a spoon. Okay, so here we have. This is actually, we often call these geranium, but they changed the name of it in the 1700s, and we're just very slow at getting used to that. Okay, so what you're best to do is to measure out quantity. So here we go again. Okay, so what you want soil, but you don't want big stones. So we just leave those out. So you've probably never seen anybody stick a spoon into a pot plant before. I use just ordinary cheap spoons as big spatulas, because spatulas are often never big enough. Okay, so let's just say we got, as I say here, we got about, take out that stone. You don't need a lot. I'm just going for around 50 grams. Yeah. Now loop. Okay, we'll come back to that pot plant in a minute. All right, Oh, so I said it used to be called geranium, but it's actually it's member of the geranium family, but it's actually called a pelargonium. Okay, that's the kind of things that you'll see in your granny's house. Okay, so okay, so that's the preservative. Just a little bit at the bottom. If you're in a warm lab, that'll evaporate. So you'll will need to check it in the meantime. But we will lower this down. Okay. Now we put in our 50 grams. We will get some soil material going straight through, trying to get all 50 grams in. Okay. So just bring that over, show you. So this is one of your experiments set up. So that would be one of your mandatory experiments, okay, in the ecology section, to study an ecosystem. And you can do it from the comfort of the lab, especially if you get one of the kids to go out and get a cup of soil. Okay, so that's number one for today. However, something I like to do as well, okay, again, it comes under the mandatory experiment of studying an ecosystem, is, well, what about something else? Because that was just arthropods. We just don't have arthropods in soil, and that's for those of you who aren't kind of thinking arthropods, spiders and insects. In soil, there are micro spiders. We'll have a look at I'll take photographs of the results, and we'll see, hopefully we'll see something, okay? And if not, I have ones from before. So a different version is if we're going to look for, this time, roundworms, okay? Normal component of the soil ecosystem. All right. So this time I got a little piece of muslin. Okay. You can usually, the reason I use muslin is because you can see it's got holes in it. All right. And I'm going to weigh out. Where's my weigh boat? There we go. I'm going to weigh out again 50 grams. Okay. Weigh out 50 grams. So tare that. If you hear me saying tare that, it's T-A-R-E. Yeah. Pick out the little stones. We don't want that. I can top up the plant, by the way, another time. So this is actually the topsoil from here. When I say here, I mean the campus. If you have lots of nematodes, that is roundworms, is that good or is that bad? What do you think? Take out the stone. No thoughts, people. Put it in the chat there. Just go yay or nay. Good? Yay or nay? Good to have roundworms? Yay or nay? $50 grams. Okay. So something intuitively, some of you are going, yay. All right, good. And you're right. But of course, it depends on what sort. If your soil is full of parasitic roundworms, maybe not. Okay, but most roundworms are not in soil. And what do roundworms eat when they're at home? Well, roundworms will actually eat bacteria. That's what they do. Typically in soil. Okay, so put that down again. So what we have, we'll hoof that on there. Okay, and we make a little. Okay. We make a little sachet, as they might call it, a little thing. We just, I don't know, did you see me tying that right? No, you probably didn't, sorry. Have to be in two places at once. I need two cameras. Okay, there we go. All right, 50 grams of soil, okay, fresh enough soil. Oh, come in, Sally. Right. This one. We have this here, and okay. Okay, so if a funnel, no, not a champagne glass gone wrong. Okay, and at the bottom of the tube, I'm going to use two clips. Two because I never quite trust them. I'm going to close the rubber tube, the length of rubber tube that's there. Okay, put that in the middle. These are two types of clips. One of these is called a Moore clip, and the other one's called a Bunsen clip. And I'd have to look it up to remember which way around it is, but it's not important. Okay, so two clips. So I'm going to put water in that. Great. So now, so we're going to set up a second experiment. It looks similar in some ways. It's probably easier if I take out the water first, Tom. Anyway, I tested it. I'll come back with the water. And we put that straight in there. Okay. There. Okay. Oh. Mm-hmm. And this is the thing. Now, what we want is for the, so you can see it's quite similar, but we're not going to put a gauze in here this time. We're going to put water. What we do is we just bring it up to the water touches the bottom of the little sack. And of course, it's going to absorb into the soil, and that's going to draw out the small beasties, the ones that we want. And the ones that we want, as I said, are nematodes. Okay. So we can raise up by pulling the light up and down. So there we go. This time we're using this with soil. Of course, this is actually medical and a veterinary procedure, just over 100 years. And it was used or discovered originally that this is how they looked for nematodes in another context, in the medical context, in the veterinary context. So that's kind of gross. So instead of soil in here, they would have been putting stool sample, feces of animals or humans. And the original worker was a fellow called Baermann. What that one is. So there's two systems for looking at soil organisms. Okay. Now, of course, the third thing you can also do is, and it's from the first week of the practical work, you got your agar plates, and what you can do is make up a slurry of soil in water, and put in your loop and streak plates, and you'll get some soil bacteria. So you can look at the bacteria, you can look at the nematodes, you can look at the arthropods, all right, as well as then things like earthworms. Okay. Now, the bigger things that you get in soil, earthworms, you want to check that you're not kind of being cruel to anything. And earthworms are at the size, they're the size of the animal really that we would not like to be hurting or damaging in any way. Okay. Anything smaller than that, not quite that it's fair game, but we have less of a consideration because as far as we know, they don't experience pain and so on. So we're not worried about nematodes feeling pain. Okay. But we do with earthworms. Okay. And they are very sensitive creatures. So if you don't want to be handling earthworms, for example, too much, because the salt from your skin actually burns them. So that would be quite traumatic and causing pain for them. Okay. But so I would check when I'm taking my soil sample that we don't have earthworms in it when I'm doing this here. I'm not worried about microspiders, you know. So there's two things. You got the microbiology, and then, as I said, the wormery. Okay. I'm not going to set that up today. I thought I'd do it next time because I'm going to actually bring some worms in and so on. And then, of course, in another time, I might actually do a dissection of an earthworm. Now, you might say he's just worried about cruelty and so on. Yeah, but as far as I'm concerned, dissecting one is better than, say, accidentally killing one and causing it lots of pain. There is a way to euthanize it without actually hurting it too much. Okay. And why do we do that? Well, I suppose the reason that you would, for example, look at dissection of an earthworm is to look at its reproductive system, its blood system, its nervous system. Okay. And the remarkable thing about an earthworm is that it has a very low differentiation, very sort of minor level of what's called cephalization. That means a head. There's no head in an earthworm. There's no, it doesn't have eyes. It has eye sensitivity spots. It doesn't have a brain. Okay. And yet it has a lot of functions of a higher organism. So, for example, an earthworm can have memory, for example. It can remember pathways in a maze. You can set up a maze for an earthworm to remember where the nice soil is, take it back out, put it back at the start of the maze. And after a number of goes of doing this, it will actually remember. It doesn't have to keep trying both channels or several pathways. It'll remember and go straight to where the soil is. Now that implies a memory, but here we have an animal that doesn't have a brain, let alone a head. And how on earth does that happen? And of course, it tells you something about yourselves, and that your spine actually takes on a lot of the functions that you think are associated with brain. Okay, so and as we know, disruption to the spine in humans is usually catastrophic. Okay, so as with a lot of things, I look at plants, we look at small beasties, we look at a lot of little organisms and so on. Why do we do that? Why can't we just learn about humans? But you see, you are learning about humans. That is precisely why we do this, because we cannot bring a rock a human in here and start dissecting. Okay, so we can't do experiments on humans. Okay, the reason that we look at these things in biology, in our Leavenscert biology and so on, is because we learn something about ourselves. That's the reason. So we look at the nervous system of an earthworm. We look at the evolutionary history of ourselves and where we've come from. We look at the DNA, for example, in an onion. We are trying to understand the information framework that how we function. Okay, and so on. Okay, so that was section 1. That's a bit of ecology, believe it or not. Okay. Behind me here, I have another station. Okay, and this time, what is this station? So what I've got here is I've actually got some of my teaching resources for when I was a teacher. Okay, and some of them aren't, but some of them are the same. Okay, these are, you can see here, drawings, you know, and those would have formed handouts. Okay, you know, simple drawings there of how to tell plants apart. Here we have the human here we have human reproduction. It's a drawing of sperm and ova and fertilization and early zygote embryonic development. Here we have, that's the colored one, but here we have then a chart on the evolution of plants. That's just the color version of it. Okay, here we have a plant model, a flower, just a flower. Okay. Here I have a dissection of a flower. Okay, that's a dissection of a lily. Here we have a coloring in sheet of coronavirus. So that's obviously recent. A model of a bacteriophage, whichever way you prefer. Okay. There's a model of DNA. Okay, you can see Shorty over there. He's the torso guy. He's just known as Shorty. Okay, and Ivan's the skeleton. He's not a real skeleton. The school I went to, which shall remain nameless, had a real skeleton, which is kind of icky. All right, but all plastic and all medical skeletons are actually from the same original mold. All of them. Which is kind of weird because it means that some guy, their skeleton was used to make a mold and they're immortal, effectively, because you know they've been copied millions of times. And it's supposed to be some Russian guy who was 17. It begs the question: did he give permission? And is his family being recompensed for all the times that his skeleton has been copied? What do you think? Okay, so yeah, so there's actually a funny, you might think the ethics of an earthworm, blah blah blah, but you know, here you have the ethics of a human, you know, being discussed. So what are all these things that are here? They're obviously learning resources, they're obviously models. Okay, and I suppose you know, more than say physics and chemistry, models are used. Now, these are what I call physical models because they're obviously mental models or theoretical models as well. So these are physical models. And we can use them for demonstration. We can use them as discussion, to set off discussion and so on. Not for filling time, we get for attracting attention and so on. The model, the anatomical model I had in Ashburn was it was actually a full-size one, and of course this one is actually from around 1970, so it is quite old now. Okay, what you can see here, and of course the advantage is you, particularly when you're working with a diagram of the internal anatomy of a human, which is totally, it's a model in itself. A diagram is a model. It doesn't tell you anything because it doesn't tell you position, it doesn't tell you size, and it doesn't tell you shape of anything. It's a very bizarre thing when we do a diagram of the digestive system and so on. Okay, because we tend to just do a kind of like a few S bends to make the small intestine. Okay, we tend to make the stomach like a sack. Okay, and then we just have a tube, the esophagus, going down. Okay, of course it's a lot more complicated than that. All right, you can see here, for example, that the colon, the ascending, the traversing, and the descending colon, you know, look at the color of it. Look at the fact that it's constricted. you know, periodically. Okay. Look at the large, you know, blood supply going to this is the stomach here, whereas this is the liver, of course. Okay. And you can see that the stomach is high up on the left-hand side. Okay. And but typically, you know, if you've got, say, indigestion as such, you feel it more or less in the center, which is why, you know, often indigestion can be misinterpreted for some kind of heart difficulty. And again, when you look at the heart, of course, it leans to the left side, as we have here, and these large blood vessels immediately radiate on the left side, which is why, you know, pain going down the left side, you know, indicates heart issue. Okay, and so on. Now, the problem with this model is that the reproductive parts are missing. Okay, so this is not a human, it's some particular version of a human. Okay, in older years, the thing that gives trouble with some people is what we see here. See a little green thing here? That's the gallbladder. Okay, so this object, this model, it's there in terms of biology, it's there for discussion. It's there to initiate discussion and so on. There isn't anything much more we can do with that. Okay, you could, for example, in a practical session, you know, try, you know, have the separate parts, you know, laid out and give them to people and say, well, what is that part, and so on. But do they need to know that level of detail? I don't know. Okay. Similarly, you know, when it comes to bones and so on, and you'll be able to probably, how you doing, Ivan? You'll be able to, you know, obviously know probably all the names of these bones, blah, blah, blah. But do we need to teach them in the Latin? Okay. Do we not need to just go collarbone, shoulder blade, and so on? Why does it have to be the Latin? Sorry. And you know, like, and that's a question. Okay, and I'm leaving that as an open question. All right. So you know, I suppose in the terms of the skeleton, you know, what you've got is a few things that are not like a real skeleton, and only if you've seen a real skeleton do you realize that. And the first thing, of course, is that this is all the bones are joined together. Okay, whereas in a real skeleton, it's not there. It's a jumble. You know, if you dig up a skeleton out of the ground, the bones are all disconnected or disarticulated. Okay, these have all been put together, you know, with wire. Don't know, can you see it there? You know, hold together with wire, pieces of wire, nuts and bolts, you know, and so on. Oh yeah. Okay, lots of metal work in order to keep them together. All right, and he's got a big, you know, he's got a dirty big kind of steel rod stuck up his spine as well, which can't be too comfortable. But and it goes right the way up through his cranium. Okay, so and the minute I do that, you can see the skull rocks forward. It can't hold itself in place. Why? Of course, because all the ligaments, tendons, muscles are not there anymore. The other thing that's different is that this is what bleached bones would look like. Okay, although they'd even be whiter. Bones aren't white typically. When if you've ever had a bad break of a bone, I was leading a biology field trip on the seashore, leaving cert biology field trip, and of course I slipped on the rocks and split my knee open. And I'm kind of looking, should I really be able to see my kneecap? That cannot be good. And of course, but what color? And of course, as a good biologist, I'm interested to see what color is it. And of course, it's got an almost translucent appearance, and it's clear you can see a kind of a range of pinks in under the surface of the bone. Okay, and when bone dies, okay, because it is a living material, it goes a brown color. Okay, and when bones are dug out of the ground, they're not bleached white. Okay, so it's not like Halloween. All right. When you see, so when someone robs the arms and the legs and the skull off your skeleton, and then you see them in someone's front garden poking, you know, sticking out of the ground, you as a good biologist, you go, well, that's not accurate. Okay. And then you can take your skull and legs and arms back again. In the plant one, you know, it's also, again, what I'm trying to do sometimes is challenge the preconceptions that people have about plants, okay? That all flowers look like the picture in the book, which they do not, because the picture in the book is a model. One of the things I wanted to do is experiment on looking at extracting chlorophyll. Okay, and basically it's a follow-on from the junior cert. If you remember when you did the separations in junior cert science, you've got distillation, you've got decanting, and so on. And you might also, you know, talk about a thing called chromatography, and you might demonstrate that with some felt tip on some filter paper and drop drops of water on it, or you might do it with the pigment of Smarties and do that. So it's simple little kind of kiddie experiments. But when we are in the Leaving Cert, where we have pigments that we're interested in, and they're the pigments in plants, okay? And what we want to have a look at, for example, is the pigments here. Chromatography, we're going to use paper. So it's chromatography paper as opposed to filter paper. You can use filter paper in the junior cert, okay? So here we have a piece of chromatography paper, and it's long enough to go in here in our chromatography jar, okay? It'll actually would sit up there. We have a glass rod that we attach it to, so we usually just fold that over. And on the bottom end, okay, that'll work nicely. So we'll make up the solvent now in a minute. I'll just show you what I have here is some pigment that I've extracted from geranium, you know, so, or pelargonium. Again, how we do that is we just collect some leaves off the pot plant. There's the geranium plant there. We put it into a mortar. Here's the mortar and pestle. If their pestle goes all a greeny color like that there, that's, you've obviously got the pigment out of it. Okay, you then can use any solvent to get it. So usually alcohol is our go-to one. This happens to be isopropanol, which is what I have here. So that's grand. Then we have to use the Pasteur pipette as a dropper, but it's not quite, it's too wide. The other thing we need then is a hair dryer to dry our spot. Okay. So that's it. So what I'll do is bring up the method. Hopefully I have it up there. I was just going over it. Again, you know, when you're a teacher yourselves, you'll be going over the practicals the day before. So here we go. Okay. Share. So there we have, so I think I've linked this in Loop. Okay, so it's chromatography, it's ascending. In other words, it's going up the paper. It's two-way because we're going to change directions midway, and we're going to separate plastid pigments. Plastids are chloroplasts and other objects inside the cells which contain pigments. We need a chromatography tank and a lid. I just use a tile or a piece of glass for the lid. Chromatography paper. I have 25 centimeters long, but it's not centimeters wide, so I'm going to change that. Pasteur pipettes or capillary tubes, some kind of clip or staple, grass, other green plants with carrots and tomato puree, whatever. Solvents, acetone, and that 1090 by volume, chloroform ether 3070. You have to set this up in the fume hood with full PPE. So we're going to use 50 mils. So I make up about 100. So we're going to go and make these up. Okay, I make them up only fresh because I'm only going to do a small quantity and I don't want them sitting around. So it's there's our ether. So we got ether, acetone, 10 of ether to 90 of acetone. Okay. Ether is the one you really have to tighten the top of because it's a very pernicious solvent. Okay, that'll do. It's a little bit shy and meniscus. Okay, so that's our solvent. So we have our solvent. Now I'll show you how to make up dropping. Okay, now move the solvent away. Don't. Okay. So, can you see this as well? I'll stop sharing. That one. So we've got the Bunsen going here again. Turn the flame quite, a good bit down. Now, if you aren't watching, you won't see it. So you pull out the Pasteur pipette and you break it. Yeah, that'll do. You just break it off, and that gives you your dropper. Okay. I'm going to use some of the solvent in the mortar and pestle just to agitate up the okay. There's our piece of paper. Now that's grand. So just make a ruler mark about 2 centimeters on the bottom. X, I'm just going to do 2 and you're going to dry that. So you can see so, a few tips. If you use ordinary filter paper, what happens is that you make a hole in it really easily. It can't tolerate this kind of treatment. Chromatography paper has a different structure to it. Okay, so the idea then is to get a green spot. Okay, usually on the X. So the more you do, the better the result. And eventually, okay. Now that's that one. What I'm going to do then is just use the pigment I made 2018. Mhm. It doesn't look to have a good, yeah. Mhm. Yeah, right, well that's making a mess. We'll go with that. So next thing, chromatography jar. We just add our pigment or our solvent. Okay, and we make sure our paper touches it. Okay, there we go. So that's chromatography, and I don't know, can you see, but immediately we can see the pigments on the freshly made one. I didn't have the patience to do the other one. I would have to make a whole load of droppers. Yeah, but I want to turn it sideways with another. So we'll let that run for a while. It's rocketing off there anyway. Okay, so you can hopefully you can see that there's a very fast kind of result. Okay. The reason the other thing is the reason I choose the geranium slash pelargonium is that it has usually a good mix of pigments, so you tend to get a good uh, okay. All right. Next thing then is going to do. You might have done. Have you done thin layer chromatography before? You probably did HPLC and all of that, but you're not going to do that in school. So this is, instead of chromatography paper, This is a glass plate, and it's coated with silica gel. And again, we can do the same thing actually. We can take our exactly the same, okay, and we can just spot that on there. Exactly the same. Okay, and you can see it's easier. You can just pop that in there, and we'll see what happens. Immediately, it's done what it should do. Okay, so we'll have a look at those in a little while. With the paper, though, you have an advantage, and the advantage is that in a jar, you're not. You can rotate the paper 90 degrees, and you can separate then the pigments in 2 dimensions. Okay, or yeah, it's usually called, when you do that, it's called 2D chromatography. It's a little bit more advanced than just your basic junior cert kind of thing. Okay. Okay, we're going to come back to that now. One of the mandatory experiments then, so that's taking chlorophyll out, which is kind of an important biochemical. All right, but it's taking out lots of other things, and we're trying to separate those out. If I just take out the chromatography plate, you can see immediately as he dribbles solvent over his computer, you can see there the line already starting of the pigment coming out. Okay. Okay. So now here, okay, you can see we've got an onion, and we got this thing, and we have this packet of stuff, and we have these. Okay, and again, where's my geranium slash pelargonium? Okay, this per plant is getting it in the. He's going to get done today, as it were. Okay. So one of the things is about extracting DNA. Okay, and uh, I'm going to show you 2 methods here. I'm also going to run through another method asynchronously, so you won't have to worry about it. We'll do that after I let you go. All right. But in the meantime, this is what we've got here. Okay. So you can see these are called in the packet DNA extraction cards. It's a peculiar thing. So it's a whole new thing for me. The chemicals for the extraction of DNA are impregnated into the card. Okay. And what I've got is I've got a native Irish geranium that I collected yesterday. Yeah, I mean, I'm thinking of you people at the weekend. Yeah, I know. How weird is that? All right. Now, what I'm going to do on that, okay, this is just this is a biopsy puncher. Okay. So, heaven thinks how we're going to do that. So, I get a leaf. I'll demonstrate it with this leaf first. Okay. So, we get it here, and we take a punch. Okay. Okay, so the trick is not to go too hard on the thing. Pop out the There we go. And there's some discs. Don't know, can you see those? Little tiny discs of the leaf, and scrunch them just on the impregnated paper. Okay. Okay, don't go too hard at it. Okay, just that's it. That's one. I will just have to change this. So what I'm going to do is get a leaf of the wild native geranium plant. Yeah, I'm doing it on the card this time. It's probably not what you're supposed to do. Anyway, okay, fish that out with a bit of wire. Yeah. Okay, and again, scrunch it. Okay, into the paper. Uh-huh. Okay. All right. Okay. So, those of you who don't know, this is this wild geranium. Sorry, it's not a great example. There we go. There's the flower. It's known as Herb Robert. It's wild geranium. So what we've got is we've got the pelargonium, which is our lab model. It's another model, by the way, a living model. And we've got our geranium. But what we're going to work out is are they really related to each other? Okay. So we should probably do a plant that is not related. So we'll just get a few bits of rose. Okay. And that will be like a control. Okay. And again, just agitate the tissue sample. Okay. And okay, so we've got right on it then rose. Geranium, Robertianum, and then pelargonium. Okay. And blank. Okay, so that's one way to get. So we're going to leave that to do its work, and then when I see you again, we'll see. Well, what do we find? Okay, so just to recap, so we've got wild geranium. Okay, and we've got this the pelargonium, and they're in the same family of plants because of the structure of their flowers. This comes from Ireland, but all across Northern Europe into Asia. Whereas this is a garden variety of a plant that comes from South Africa originally in the 1700s. So will we see that in their DNA? That's the question. Will that be obvious? Okay. Right. Now, you look at your biology textbook, though, that's not how you're extracting DNA. And as well as that, you're not going to look at it and do anything with it. But that's what we're going to do. So we got an onion. All right. So guys, here's a question for you. What part of the plant of an onion is the bulb? What part of the plant of an onion is the bulb? And I want you to just put that in there into the chat. We'll see what you think. What part of the onion is the bulb? Yeah, well, this, the thing I'm holding. What part of the onion plant is the bulb? Okay. Okay. All right. Now, what we take these are usually sticking out of the ground. Okay. No. Uh, that that at the end, that's the roots. Those things there are the roots. And because they've all dried up, you don't see them too much. But the roots are here at this end. And then out of this pointed part, you'll have a green tubular leaf. Okay. So if you've got roots here at this end, yeah, and you've got leaves coming out of here at this end, what's in between? It's a stem. So this is a modified stem. All right. So, if you ask, what parts of the plants do we eat? Aliens came from abroad, from outer space, and they said, okay, what parts of plants do you eat here? I don't know why they would have American accent. Um, you would say, oh, well, some plants we eat the leaves, sometimes it's leaf stalks, sometimes it's the flowers, sometimes it's the fruits, and sometimes it's the roots, and sometimes it's the stems. So that's a modified stem. It's a short stem, but all bulbous plants have short modified stems. So let's say this is the practical as it's in the textbook. We'll try and work a bit faster, Tom. Okay, so we'll just butter. Okay, so we cut an onion up. Doesn't have to be the whole onion. Now we could use a food blender, but hang on, I'm going to use again mortar and pestle. Okay, and I'm just going to add a little bit of salt. Yep, sodium chloride. The minute you do this, you're amazed to see how much liquid suddenly appears. Okay, so question number one is, what does the salt do? It relates as to where the liquid is coming from. Okay, good, good, good. I'm just putting that into then a small beaker. Your test tube, it doesn't matter. Okay. Okay. Next thing I add is then some washing up liquid. Add a spoon of that. Okay. Super. And then a little bit of water. Okay. Let's leave that to sit for a little while. Okay. And then, this is what I did earlier. I put some alcohol in the freezer. Okay, so this is alcohol that's been in the freezer. Okay, and it's neat. Okay. So we've left that just for a couple of minutes, and we drizzle that on the side. Now, freezing alcohol doesn't mix so easily with water. Okay. And then what we should see, let's just make that horizontal. We should see strands of material rising up from the the aqueous stuff at the bottom. Okay, and that is supposed to be, well, at least have DNA in it. Okay. Okay. That is, believe it or not, that's the positive result. Okay, now, what if we wanted to stain the DNA in order to prove that that's the DNA? What would we be doing then? Okay, what's the stain for DNA? There are several stains, and they're usually a derivative of methylene or methyl blue. Okay, if you're staining animal cells under the microscope, methylene blue is what you're using. Okay, so we could add a blue stain. The alcohol would go blue because you're diluting it, but what we would be looking for then is that you would see dark blue strands or clumps of dark blue in there. But methylene blue also stains protein. And of course, the DNA is completely bound up with protein. So, you know, you're getting a lot of protein material in there as well. So the way around that then is to add a protease to this. Okay. Right. So you've seen one of the two more kind of, I suppose, recent in that they're the last 20 or 30 years they were developed. Okay. Now that's the, if you like, the kitchen version of extracting DNA. Okay. They typically the way that they do it is the chemicals, the same kind of chemicals. So think about it. You had a solvent, the last stage. The second last stage was a strong detergent, and the first stage was something to lyse the cells. So we burst the cells using the salt. Okay. We then broke up the lipid membranes with the detergent, and then we precipitated the DNA out with freezing cold alcohol. Okay. So there are more kind of, I suppose, technical ways to do that, but those are your three kind of steps. Okay. So it's not just a matter of repeating this in a biology lab. What it is trying to understand, well, what are you doing? Okay. So you've got to break down the plant's walls, then the plant membranes, bursting out the DNA, because remember the DNA is bound inside membranes in the nucleus. And the step that I could have introduced there would have been to use a protease, but I didn't. And then precipitate out, because DNA precipitates in freezing alcohol. Okay.